Welcome back to the Pager podcast. In this episode, we take another deep dive into a long-standing interest of mine, which is evidence-based medicine. My guest today is Professor Paul Glasziou. Paul is the director of the Institute for Evidence-Based Healthcare at Bonn University and really a titan of this field. He is also the former director of the Centre for Evidence-Based Medicine in Oxford. Some of his particular interests relate to reducing research waste and ensuring that high-quality research actually translates into clinical practice where appropriate. We spend a lot of the episode talking about these topics, from discussion of publication bias to design of research, incentives for researchers, and how we should be interpreting evidence in the context of previous research, some of which might be conflicting with new research that's emerged. We also talk about how much evidence should be needed to change practice, and we discuss weighing up whether a body of research can or cannot necessarily be applied to making a decision about an individual patient. As ever, if you like this podcast, do share it with a friend, come back to listen to our other episodes. We would also love to hear your feedback, and you can reach us on social media and at pagerpodcast@gmail.com. Brilliant. So, Professor Glasziou, thanks so much for joining me from Australia. We're in slightly different time zones at the moment, but it hasn't stopped us connecting, which is good.
Thanks very much, George. Pleasure to be here.
So, just to start off with, can I ask what your interests are and what your research interests are in the area of evidence-based medicine?
Right. So, I've spent most of my research career working on how to improve the evidence base of research, but also how to get clinicians to better understand and use evidence. This was largely driven by my own experience in clinical work initially and then in research, and then seeing the disconnect between the two, and was inspired by the group from McMaster who founded evidence-based medicine. That's Dave Sackett, Gord Guyatt and others back in the early 1990s, and inspired me to go back into clinical work. And then I worked in emergency rooms for a while, and then in general practice. And so living in those two worlds, what I'd been wondering about is how do I make research more relevant to practice, and how do I get practitioners to understand the relevance of research, trying to get the two worlds a little bit closer.
And I'd like to take, I think, the first part of that to begin with. And I think intrinsic to this is something you've written about wasted research.
Sure.
And could we perhaps go into some of the ways in which research can be, I guess, wasted and why this might be the case?
Sure. So maybe I can tell you how I got interested in this area is that I used to be the editor of the thing called the Evidence-Based Medicine Journal, which was the brainchild of Brian Haynes, one of that McMaster group. And what he was doing was scanning the world literature, which is pretty big, obviously, trying to identify those new pieces of research that should be influential in clinical practice. You know, a new big trial comes out, or a new diagnostic test, and this is the definitive study. And he would then write one-page summaries of those with a clinical comment. So the American version of that was called the ACP Journal Club, which is still running, and the British or European version was the Evidence-Based Medicine Journal. Fantastic resource. But one of the things that I came to recognise in running that, in editing that resource, is that there were a lot of treatments that were being promoted, that the research had shown was effective, but I couldn't do them in practice. That was particularly true for non-drug interventions, like salt reduction, being shown to reduce cardiovascular events. But how do you get a patient to reduce salt? Turns out you can't just say, well, reduce your salt. You can't even simply say that it's not just the salt that you're putting on your food, it's also the salt in the tins and cans you buy, the produced food. It's actually quite complicated, and the trials that have been successful often had sessions of 10 to 12 group sessions with a dietitian in order to get people to reduce their salt just enough to lower their blood pressure a bit. But that was incredibly complicated and not well described in the papers. So we then did a study to look how well interventions are described and discovered that less than 50% of them are sufficiently well described that me as a clinician could have just picked them up from the papers and used them without writing to the authors and getting more details. Isn't that a waste? You've done all this work. You've published the paper and revised it and got it in the journal. Fantastic. And now nobody can use it.
That's incredible. And I guess there are two things there. There's firstly, actually, is the intervention feasible with the resources that you have in the current moment? But also something I think even more incredible, as you just said, do you even know what the intervention is, and can you replicate it yourself?
Yes, that's right. So that was part of what became a bigger picture. So I was working with Ian Chalmers, who was the founder of the Cochrane collaboration, and he and I were regularly discussing this. And we recognised that it wasn't just a problem for the description of treatments, for example, but there were lots of other problems that made research unusable that was avoidable. So we wrote this thing about the avoidable waste in research that was in the Lancet in 2009. And that was me trying then to get the researchers to move closer to making things good for the clinicians. But at the same time, you've got this other problem that I mentioned of the clinicians needing to move closer to understanding what goes on with the research and what bits of it are usable.
And I'd recommend everybody to, I think it was 2014 that series came out in the Lancet, and it's a very interesting and very accessible read. So there's a plug for that there.
That's right. And if you want a much more digestible version of it, we also wrote some BMJ blogs that make it a lot easier to understand what was in that long series in the 2014 Lancet series.
And one number that you arrive at is 85%, I think, in that series of research being, in inverted commas, wasted in 2014. What were the main components of this? What were the main culprits?
So one of the BMJ blogs walks through that to explain exactly how we got to 85%. But the simplest way to explain it is that 50% of clinical trials never get published or reported. There's been some improvement recently because of trial registration, but that figure had been pretty stable for a long time. So that's where half the waste comes in, is research that gets done that never actually gets reported. The second element is what I was just describing about, that the reports that come out are unusable because something is missing from them. There's not a complete description of the outcomes of the intervention or the nature of the treatment. So that's the second, and these are multiplicative. So that's a second 50% wasted. And then the third was that we could document that there were avoidable design flaws, studies that could have been blinded that weren't, or studies that could have been properly randomised that weren't, etc. And if you put those three in sequence, the 50% is not well designed, then 50% of that is then not published, and even when it's published, 50% of that is unusable. Multiply those together and you end up with 12.5%, 0.125, being usable, right? So we rounded that a little bit to 15% being usable, or 85% being avoidably wasted. There's, of course, waste in research because, you go down blind alleys that you can't predict. Take all of the drugs that have been tested during the pandemic. In particular, the recovery trial has looked at maybe 10 treatments or so. Most of them have not worked, but a few have. That's not waste. That's finding out what works and what doesn't, which is the nature of research. You can't know what you're going to find before you start. So we're not talking about that. We're talking about avoidable waste because of poor design, non-publication, and poor reporting.
And I suppose there is this balance between, taking everything to trials and then having a very small number of successes, and taking not enough things to trials where actually perhaps there was equipoise before, and these would have benefited from being studied. So finding that balance is quite important.
Yes. Importantly, though, there's more and more research happening. So the numbers of trials have been growing exponentially since the first trial, which was in 1948 of streptomycin for tuberculosis. And then trials got a big push with an act that was passed by the FDA in the United States back in the early 60s because of the thalidomide disaster, saying that you wanted to prove that there was benefit using trials because of the safety concerns. You wanted to make sure at least that the balance of benefits and harms were in favour of benefits. And since then, you've had this steady increase so that we now have tens of thousands of trials being published every year, or hundreds of thousands, in fact, now. And it's increased the range of things. Initially, that was really just drugs, and there was the big polio vaccine trial. But now we're doing it for non-drug treatments, for diagnostics. Even changes in the way that we run health services are now sometimes being put to randomised trials.
And we've seen trials of non-drug interventions in the pandemic be quite influential and really, I think, highlight the importance of not just focusing on these drug treatments.
That's right.
And I'd like to dive in a bit more into talking about this lack of publication and actually reporting of results, because I think it's a real issue. And I'd perhaps like to turn the table around and say, are there any scenarios in which it is reasonable for research not to be published in full? To play devil's advocate here.
So just one clarification. I think every study that gets completed should have some sort of report that's publicly available. That doesn't necessarily have to be in a journal publication. So one alternative that has happened, because there are things called trial registries now. So in a lot of countries, in order to run a trial, prior to starting the trial, you have to register it. You have to say who the patients are, what the interventions are, how you'll measure the outcomes. In a sense, a summary of the protocol. And then you do it. But those registries are now open to people posting the results. And in fact, in the US, it's mandatory to post the results of your trial in the registry. That's actually meant an increase in the number of study results or trial results that have become available because they're being posted in the registry, even if they're not being published. So, but I think it should be mandatory that every trial where you've started patient recruitment should have a report that says, here were the results. Even if you stopped after 3 patients because of a safety concern, that may be important to somebody else because of the safety concern.
I think there are a number of cases where that's been highlighted, that actually more information that was not out there in the public sphere would have been very useful for planning future research. Those, whether actually it's a safety signal or whether it's simply a dead end.
Yes, exactly.
Another way in which it serves to, I guess, detriment patient populations is by skewing the direction of the results in published research. And I was wondering if you could tell me a bit about that.
Yes, so that's the problem of publication bias. So even if there was no publication bias, you've still got this waste in half that the study's never being reported. But it's even worse than that, in that the ones that we have access to tend to be those that are more likely to be statistically significant or positive, so-called positive trials. And the half that we don't get tend to be the negative studies. And that was demonstrated now about 30 years ago. A couple of people looked at the studies that were registered or had passed through ethics committees and then followed them up to see what got published. And one very interesting thing that a woman called Kay Dickerson at Johns Hopkins found with her early study of this is that the problem was with the authors. That is, the people who had conducted the study didn't submit the results. It's not because the journals were rejecting them, which is a common thought of publication bias. It's because the journals are rejecting them. It's not. It's that the authors are not submitting them. And the reason for that? Well, it's lots. Sometimes you've got several studies going on one has an exciting positive result and the others have negative results, which one are you going to get all excited because you think you can get it in the Lancet, right? So that's the one that you'll prioritise. And so there's either a delay or sometimes non-publication of those negative studies. But other reasons that can happen that we've seen, even for very big studies, is that people have retired or even died without publishing their reports, or they've moved on to another job. So actually we need some way that when a study is completed, that there's a clear sort of chain of responsibility, that if the lead author moves on, decides to go into politics or the circus or whatever, that there's somebody else who's responsible then for making sure that that gets published. Ultimately, that should be the institution, of course, but actually you need a chain of individual responsibility as well.
And that's really interesting because I think perhaps some people turn around and say, okay, it's because these things are not being published that they're not being available. But actually, we've seen mechanisms by which data can be available if you want it to be available in reports. For example, just putting it on this trial registry here. And the people in the top journals would probably turn around and say, we do publish negative results, but I think it's fair to say they are published less frequently and they have to be of higher caliber perhaps of research in order to compete with the positive results. Is that fair to say?
Well, yeah, and it's also the level of the journal. You probably won't get your negative results unless sometimes it's a very interesting negative result. And so that can get published.
If it changes practice, for example.
But a lot of negative results are sort of uninteresting and therefore they get published in lower tier journals. And so if I've got one that I think, I can get this into one of the top tier journals and this other negative study that's only going to get into a low tier journal, you prioritise the good study, the positive one, sorry.
And this, in fact, links in with another one of your recommendations, that actually it's almost inexcusable before carrying out research to not do a proper review of the literature and a proper up-to-date systematic review or meta-analysis of what has come before the trial. As somebody who's had a lot of experience in this area, how do you factor in this publication bias when trying to get a balanced view of what a number of studies say on a particular topic?
So I think there's two issues in there, George. One is the need to do the systematic review, but then there's the problem of there potentially being a bias because of the non-publication when you do your systematic review only on the published studies. So let me deal with the first one of doing the systematic review. I think it often doesn't get done. People initiate new primary studies without looking at the previous research, or at least all of the previous research. They'll probably know about a couple of the studies, but they don't know about the whole body of evidence. And that's partly because doing a systematic review is a lot of work. It often takes 6 months to 2 years is a typical sort of time. So to try and overcome that, we've been working on ways to speed up the reviews by developing computer automation tools that help with the process. And we've now got down to what we call a 2-week systematic review. It's the full quality of review, but we've speeded up the whole process to make it feasible before you do new primary research. You still have the problem, of course, though, of that being on published literature. So the other thing is that we need to get more of the results published in registries, but you can also do a systematic review that's only focused on the registered studies. An oncologist by the name of John Symes actually showed that about 40 years ago, that you get slightly different results if you base it only on the registered studies versus just the published studies. That the published ones will in general be more positive than using just the results from the registered studies, because the results available from the registered studies is an unbiased set. It may not be all the studies, but it's an unbiased set of studies. And I think that will become increasingly important, that we need to insist that people register all studies and that all of the reports are available for those registered studies, as Ben Goldacre has been pressing in his All Trials initiative. And then we'll overcome that problem of the publication bias in systematic reviews.
And I think Ben Goldacre has a lovely little analogy for it. Is if he flips a coin 50 times, but only shows you the result of half of those, are you going to trust whether the coin is fair or not?
That's a very good metaphor, yes.
I'd like to go back just briefly, because you said that this is an unbiased look at the trials out there. I assume that relies on actually the results. Obviously, we have registration, we have publishing of results, maybe on a registry, and then we have publishing in journals. That assumes that actually there is translation from the registration to publishing of results, even in their basic form. Does it not?
Yes, well spotted, George. It does assume that you've got the results there. That's becoming more true with the US requiring mandatory reporting of results in the registry, for example, which is clinicaltrials.gov. We've seen an increase in the number of things that are available only via the registry and not as publications. So it is important that systematic reviewers look at those. But you're right, we won't get the completely unbiased until we get 100% of reporting in the registry, irrespective of whether it gets published or not in a journal.
And I think lots of research funding organisations are now moving to instigate some much tougher rules, such as withholding aspects of funding or not giving more funding to researchers if they're shown to have not reported their results at all.
That's right. And there are groups that's a rule that the NIH has in the US, for example, the National Institutes of Health. You can't get your new funding unless you've actually published the results of the previous research. They actually have fines that are available too for the non-publication, but as yet they haven't been implemented. There is talk now of them actually implementing those fines. As Ben Goldacre has pointed out, they could actually be making lots of money from implementing those fines. But even without the fines, the National Institutes of Health Research in the UK's HTA program, the Health Technology Assessment Program, their trials achieve a 98% completion or complete reporting rate. How do they do that? I can see the puzzle on your face. Part of it's through helping people through the process. Part of it's that they have 10% of the funding that is withheld until you've made those final results available. And finally, they have their own journal, the HTA reports, where you have to publish the full version of it. Even if you publish the shorter, 6-page version of it in a journal somewhere else, the complete reporting has to go into the HTA reports. So the 10% funding is probably important, but actually it's a number of mechanisms that enable them to get up to that 98% rate. But it does indicate that it's doable if we pay attention to it.
This probably links into, I think, what we briefly touched upon earlier about actually allowing people or researchers who maybe in some cases actually haven't conducted very much research before in the past, to present their ideas and their findings in a way that is useful for people who are going to be reading them, whether it's you conducting a systematic review or a clinician who's looking to weigh up the evidence for whether an intervention is appropriate for their patient or not.
Yeah, and I think there are tools that are going to make that easier, but my advice at the moment is if you're going to conduct a clinical trial, you probably want to have one experienced member at least in the team, somebody who's done it before. It's a bit like doing, open heart surgery. You don't want a completely naive team to be doing that on you.
Exactly. And I'm curious, going back to talking about this idea of a two-week systematic review, how these new kind of living systematic reviews that I think the BMJ has been pioneering fit into this. Because I'd imagine they almost walk hand in hand, actually, that if you can conduct a review more rapidly, then actually it must be easier to update a review as well.
Well, the tools that have become available for doing the 2-week systematic review do help with that updating process. But the updating process isn't. So we've been involved in some of the so-called living reviews. You usually, because it's when they say living, it's not every day. We were getting it once a month. You get a batch of new studies. One of the important tools, though, is to say, if you've got this batch of new studies, have you already looked at any of those? So being able to deduplicate rapidly is one of the tools that you need. But there are some other things that just help you with a rapid screening process as well. And most months you won't have to do any new reviewing. You'll just tick it off and say, no, there were no new studies this month. Or there may be a new study, so you've got one new trial to extract, for example. So the tools will help you do that, but we also need better structures of reviews so that it's much easier to add in the one new study. Because if you read a review at the moment, you've got to report the number of studies that you've searched, the number that were included, the total number of patients, etc. So that flows through the whole of the text of the review if you add one new study in. It's actually very easy to automate all of that, though, to say, here it is, and let the numbers all flow through the text of that. And so until we have that sort of a tool that allows us that easy updating, it'll still be difficult to do the living reviews as it is at the moment. But we're moving there. I think those tools are being developed, and it's going to make it easier and easier in the future to make it living systematic reviews.
And I wonder if the incentive structures surrounding these reviews could do with changing to encourage living reviews. Because, take an example, if a couple of years ago I published my systematic review in the BMJ, I can put it on my CV, it's there, I've drawn a line underneath it. What's incentivising me to go out and update that when actually, for example, the faculty position I want to apply to requires more different papers rather than working on the same ones?
Yeah, no, that's a very good point, George. And I think there's a lot of push now to move away from pure publication metrics for hiring and promotion decisions. So there's a number of groups looking at that, particularly in the Netherlands, but there's a sort of worldwide movement through the so-called DORA principles, and also a thing called the Hong Kong principles, which would look at other metrics and with a particular interest in the impact. So if you had one review that you were keeping up to date, you could get credit for doing that and for the impact of those updates. And that would be a much better system, because then it would incentivize exactly what you're saying, that no one wants to update a review at the moment because they don't get publication credit. But if you were incentivized through the updates and the impact of those updates, that would make it more worthwhile on your CV to then say, I've had this thing, it's been updated 5 times and it's impacted guidelines each time it's been updated.
Another point that you've made before, or certainly was made in the Lancet series, was that actually rewarding researchers on the validity and reproducibility of their results, not necessarily what is shown by them. And that seems like something that has the potential to really incentivize people carrying out rigorous research that is, I guess, validated by the rest of the scientific community and going forwards.
Yes. No, it's a nice idea. Putting it into practice, of course, is much harder because we have to judge then what is reproducible and what isn't. Some of it can be through the documentation. So there's different levels of reproducibility. The one that I was talking about earlier, which is it sufficiently well described that another person could pick up that protocol and repeat the experiment? That's one type of reproducibility. And you could reward that to say, yes, there's a full and adequate protocol that the person has posted on via the trials registry or on the Open Science Framework or somewhere where it's publicly available, and you get credit for that. Or you lose credit because you haven't posted it. One of the two. Either way, you get an academic kudos for it. But then were the results reproducible, which is quite a different thing, and that's much harder to judge because, of course, you never get exactly the same answer. Think of just the statistical uncertainty and the confidence interval when you repeat a piece of research. And so I think some of that still needs working through. But I think there are lots of those intermediate things that we can look at in order to include those in the sort of rewards that we give to academics for doing their research, making their data available, making their protocols available to other people, and then judging people by how much reuse there is of those materials. Because again, that's a downstream impact that you're having on other people's research.
How does basic science research fit into this? Or some research that doesn't necessarily have an immediately applicable clinical role, but you could imagine being perhaps more slow burning and maybe even fueling further research, or linking in with what is being done elsewhere, but might find it harder to get credit under the system you've just described?
Yeah, no, I think there are equivalents. So you have to count the impact not on patient care directly, but the impact on knowledge. So was this an important impact on knowledge? But then the same principles we were just talking about, which is, was the protocol available, was the data available, etc. And by the way, we certainly know that there are similar problems within basic science research. A guy called Glenn Begley, when he was at the company Amgen, did a study where he tried to repeat the results of about 50 basic science things that his company wanted to use as part of their development program. But he said before he used any of them on a chain of research, he wanted to at least reproduce the original findings. And he couldn't do it for most of them. I think something like 6 out of about 45, I'm not sure if I got those numbers exactly correct, were actually reproducible. And he often got people in the same laboratory to try and reproduce the piece of research. But he said what he did was make sure that they did it blinded to what the exposures were. He said blinding was the most important thing in making research irreproducible. So he said if you wanted to change anything, 3 things, it would be blinding, blinding, blinding.
Perhaps that highlights an area in which perhaps basic science research can be even more vulnerable to this, in that regulatory oversight is much stronger when looking at participation of kind of volunteers in clinical trials. So that at least provides some level of, control and monitoring. Whereas if you're experimenting on cells, that's a whole different game.
No, that's true. The oversight is less. But I think it's, I see change happening within the basic sciences as well. People have become aware of this problem. They've started to produce reporting guidelines. They've started to do systematic reviews within basic sciences. So all of the things that had happened within clinical research that started probably in the 1980s and 1990s and have sort of grown since are now happening in basic science as well. Of course, slightly different problems, so they're going to happen in a slightly different way. But they've also seen what's happened with the trials changes, and those similar changes are now being echoed within basic science as well. They'll be adapted, but they've got a path to follow that's been lit by the trials community.
Does a current focus on authorship incentivize a greater quantity of less impactful research? To what extent do you think that is true? There can only be one first author on a trial involving 10,000 people, where there can be 5 different first authors on 5 different trials run perhaps using the same resources.
Yes, that's a problem with getting multi-centre research done. But nevertheless, a lot of multi-centre research does get done, often with a group authorship then. So I think, yes, you're right that the current authorship rules have problems. Journals now are allowing, though, that sort of group authorship, having the writing committee sort of specified and using contributorship as something. I think that's one extreme end, though, that you still have the individual who's making important basic discoveries, for example, who may want their recognition. So I think we have to find a compromise between those in recognising the sort of individual innovator versus the huge amount of work that necessarily must go into a large 10,000 person multi-centre trial.
And actually, now that you say that, I remember looking through authorships and seeing, I think more and more, I don't know if it's true for the recovery trial, but something like the recovery trial collaboration as a listed author rather than one individual in some areas.
That's right. And interestingly, the recovery trial was based on the protocol from the ISIS-2 trial, which was published in 1988. and which had a group authorship devised by a guy called Richard Peto, a statistician in Oxford, who wrote the protocol for that, but had the group authorship for the ISIS-2 study, and that was the inspiration that that Peter Horby and Martin Landray had used for designing the recovery trial.
And certainly that's, well, we've talked about it on the podcast before, but that's been a standout success in clinical trial design, and something that will hopefully be influential in inspiring future high-quality research.
Absolutely. I think it's been the standout of the trials, though there have been a number of other platform trials as well that was similar. So the Solidarity trial, which was WHO's sort of equivalent to recovery, just ran into more logistic problems in getting through their ethics and governance processes. And so early on, they didn't recruit as well as recovery, but they're now starting to have a sort of catch-up process. And there was another interesting one called REMCAP, which was looking at community-acquired pneumonia. It had been set up a couple of years ago to do multi-centre platform-type trials in community-acquired pneumonia. And when COVID came along, they just switched. They already had the collaboration set up. Again, they ran into problems because of the particular countries that it was set up in. They'd sort of missed the appropriate waves, or there were no early waves. And so recovery was not only brilliant, but it also had some good luck in being able to get going in the UK, where there was a huge first wave. And they got going quickly enough and had the infrastructure of the UK trials network and the support of all of the chief medical officers in England, Scotland, Wales and Northern Ireland.
I think it certainly made the best of a bad situation. I'll briefly explain for those who don't understand, I think, my understanding of what a platform trial is, because I think that was useful then, which is the idea that you can test multiple different interventions, potentially at different time periods. So the recovery trial took on different arms of the study and ran different arms of the study, testing, for example, convalescent plasma, testing, I think, remdesivir, testing dexamethasone. So it could do all of those and compare them to a control group together in a way that could be updated as new people were recruited to it. So really cast a greater net than a traditional trial that would have just pitted one thing against another, or maybe one thing against two other things. Is that a fair summary?
Yes, that's a very good summary, George. So you also cut off arms, like the hydroxychloroquine arm, I think, was the first one that recovery stopped, showing that hydroxychloroquine didn't work. And they'd added on by that stage, I think, the convalescent plasma. And so over time, they've finished arms like the dexamethasone and hydroxychloroquine arm, and they've added on new arms in the study because they've got the trials network set up, they've got the protocol set up, they've got the ethics set up, and they can just switch things in and out.
I think I'd like to slightly change tacks for a bit now and talk about actually how clinicians interpret research, because this was the second point that you made right at the beginning, and allowing clinicians to understand how research should influence their practice. So I think my first question here, when research differs from previous research, but actually the nature of the research is a little bit different and the nature of the methods are slightly different, how should we interpret whether it's actually a new finding or whether it's just noise in the background?
So the important principle you're bringing forth here is that you need to set the new research in the context of previous similar research. So, for example, let's take a pandemic example of hydroxychloroquine. In fact, when the recovery trial completed, they also looked at the other research that had been done to say how similar was this to other studies, because of course the group of patients, the dose, etc., may differ. One of the beauties then of having both multi-centre research but also looking at different pieces of research is you can judge whether those things make a difference if you've got good enough quality research. Of course, it could be artifactual. If you've got a biased piece of research, it may look better, and it's not because the intervention is better in that group, it's because of the bias. So you have to also look at the quality of the studies before you look at whether there's differences between the results of the different studies. One of the surprising things, I think, though, is that for most interventions that I've looked at over the 30 years of my sort of research, 30 or 40 years of my research career, is there's actually not that very much variation between the effects. It's very rare to get what's called a qualitative interaction. And what I mean by that is where it works in one group of patients, but either doesn't work or is even harmful in another group of patients. That does happen occasionally. So Peter Rothwell, for example, a neurologist in Oxford, showed that carotid endarterectomy, where you sort of ream out the carotid artery in the neck, could prevent stroke in those at very high risk. But in those at very low risk, it actually caused net harm. You are more likely to get a stroke, which you can imagine because the operation itself is actually stirring things up, to put it very simply. So you've got to have enough benefit, enough risk to repay that. So it's only the higher risk patients who actually get a net benefit. But those sort of interactions where it works in one group and doesn't work in another are actually quite rare.
I think, dredging up my knowledge of carotid endarterectomy, I think one thing that surprised me was just how severe disease has to be for you to be considered for it. I think it's maybe over 75% stenosis for the balance of evidence to fall in favour of it, which is, I guess, just sitting there thinking about it, it's enormous. The idea that you have to pass such a high threshold in order to have it cleared out.
And be symptomatic.
And be symptomatic.
Yeah, you can have an asymptomatic 75% stenosis and you probably have a net harm for those people. But again, Peter Rothwell has done some clever work where he looks at multiple factors. So if you've had a more recent transient ischemic attack, the sort of mini stroke that wears off within 24 hours, that also puts you at higher risk and therefore you get more benefit from your endarterectomy. So there's a number of factors that go.
That's why we carry out trials in the first place, to tell us.
But then to get those nuances, you need even larger studies so you can look at those subgroups.
I want to actually re-emphasise a point that you made just earlier, that actually group differences perhaps are not as common as we might suppose. So perhaps if I'm taking a cynical approach, an organisation marketing an intervention may really want to highlight the fact that this group is slightly different from the previous one, and therefore we should trust this signal. But also it rather helps clinicians in when they have a clinical decision in front of them, a patient, and they're thinking, okay, whether it's in the emergency department or in general practice, and you're thinking, does this evidence actually apply to the patient that is here? And could you talk me a bit through your thoughts on how you go through that? Because it's obviously a sliding scale between the matching exactly and not matching on every single one of the parameters. You have a paediatric trial and you're trying to think, okay, what's it going to do in somebody who's pregnant, for example?
Yeah. No, it's a very important and very good question, George. So first of all, my clinical career has mostly been in general practice, so I also did quite a lot in emergency as well. And actually, they're two quite different settings. You'll usually get in the emergency setting a more severe spectrum of illness than we'd see in general practice typically. So that distinction was actually very important to me clinically. Because the general rule would be, as I was indicating for the carotid endarterectomy, is the more severe your illness is or the more at risk you are, the more likely you are to benefit. So you could assume that the biological effect gives you the same relative effect across all patients. That's roughly true, and I'll come back to that. But then if you get that the relative effect translates into a different benefit or an absolute benefit to patients depending upon their severity or risk. So the higher the risk you are or the higher the severity, the more benefit you get. The lower the risk or the lower the severity, the less benefit you'll get. And more it's more likely that it'll be outweighed by the harms, the adverse drug reactions, for example, or just the overall harm of surgery. So that balance of benefits and harms differs with your risk. That's not an absolute. There are some things where you do get a biological difference across different populations. But if you have to make an assumption, it's a very reasonable first assumption to make, that until you see a biological reason for why it should differ in one group versus another, assume that the average relative or biological effect is the same, but what's changing is the underlying risk or severity of the patient.
Okay, that makes sense. I think in my mind, and perhaps I could also add that also applies for harms and side effects as well. Potentially, those could be in some cases magnified in a high risk group. It's not an unfair suggestion to say that somebody with a 95% kind of risk of an adverse outcome in 5 years also has a higher risk of having an adverse outcome from the intervention based on their underlying comorbidities.
Yeah, though that's harder to predict. So you can get, for example, adverse drug reactions that are just idiosyncratic. Think of anaphylaxis to penicillin. Okay, in a sense, that's irrespective of the condition that you're treating or the person that you're treating. It's just this same small biological risk of an anaphylaxis. And so if you're giving penicillin for a viral sore throat, you just risk, this would be intramuscular penicillin, you're just risking the anaphylaxis.
My understanding of anaphylaxis and its effects on different people isn't great, but to what extent actually, if you are high risk, you might have the same risk of an anaphylactic reaction to a particular medication, but are the consequences of it going to be worse in particular patients who have underlying comorbidities, or is it pretty constant in terms of its risk across the board? I'm perhaps nitpicking here.
Some differences, but nothing major. Not like in comparison with the spectrum of illness that you might be getting, because you get a healthy person who's got a meningococcus, for example, where the penicillin is clearly going to do more good.
That I think is a really useful way to frame these questions, because I've been kind of grappling in my mind thinking, okay, what is a reasonable difference between the patient and the trial population, and what is something that I should begin to be a little bit cautious about?
Yeah, it's a tough question, George. So thank you for asking it, and it's something you definitely need to grapple with. But that's what I've just outlined with that, thinking about the underlying risk, but assuming harms are relatively constant but not completely, is a sort of rough beginning. Peter Rothwell's written a whole book about the different approaches to this, but what I was describing to you is something that we had set out in a paper too many decades ago to mention.
My next question, which might also be an interesting one, which is, to what extent should practice that is based on very little evidence be overturned and changed on less evidence, or in some cases very little evidence?
So overturned, we should always be looking at the body of evidence, that is accumulating the evidence from the different studies that come out. How much do you actually need then to say, yes, we now have enough evidence that we either should start doing this or should stop doing this? Is that your question?
Yes. I guess I'm thinking of a scenario in which, if you appreciate the need to look at the body of evidence, but actually if there's been so little evidence before, then changing from your default position could be on the basis of something that's actually very low quality to begin with, if you see what I mean.
Potentially, yes. So let's start with the extreme. You occasionally get dramatic effects that are so clear that you don't need much evidence at all. I'll give you my own personal experience of how I got to thinking about this in the first place. Was that when I was working in general practice, I had a very young kid, about 3 years old, brought in, and they had a plastic bead that had been stuck up their nose. And I asked one of the nursing sisters at the place that I was working if she could get me a pair of long forceps, the artery forceps, to try and pull this bead out. She said, what for? And I explained, and she said, well, have you tried the mother's kiss technique? And I said, no, what's that? And this is a very useful thing clinically. You may need this one day, George, in your emergency department. The way it works is you occlude the nostril that doesn't have the bead in it, right? And then you get the mother to blow in the mouth, and it forces air down the back of the nose through the nasopharynx. And that pushes the bead out. That's the mother's kiss, or parent kiss technique, is the ungendered version of it. So I tried it and it worked. And I thought, well, that's pretty convincing. Do I really need randomised trials to show that the mother's kiss technique works? Because of that instantaneous and obvious effect. And we get some of those. We've had a few in medicine. So, for example, defibrillation, when it was first invented back in the late 1950s or early 1960s, brought people back to life. Okay, it doesn't happen very often, so, that was pretty dramatic. Or someone who's basically blind from cataracts and removing their cataracts, and suddenly they can see again. So there are a number of those sort of dramatic things. My favourite example being this picture I saw of a guy who'd had his arm bitten off by an alligator, and then it was sewn back on again. Okay, we know what happens. We can predict if you don't sew the arm back on, it's never going to work. It's obvious. But there's very few of those in medicine. Most of the things are much more like the carotid endarterectomy that we were discussing, that lowers the risk, or lowering blood pressure for preventing stroke. It lowers the risk, but it's much more unpredictable. And for those things, we generally need randomised trials. How much do we need? Well, that's what the GRADE process, I don't know if you've heard of GRADE.
Oh, and in terms of levels of evidence?
Yeah, the GRADE levels of evidence has been working on exactly this. And in fact, they do take into account that sort of dramatic effect that I've just been describing, because that size of effect is one of the things that can upgrade your evidence. So you need less quality and less amounts of evidence if you've got a very dramatic effect like that. So what GRADE takes into account is the amount of evidence, the quality of the evidence, the consistency of the evidence, the size of the effect, the likelihood of publication bias. So they take a range of things into account on the body of evidence to say, yes, this is good enough evidence that we think you should, that it's high quality evidence to change your practice. But actually they've got an interesting second stage to that for a recommendation, which looks at the balance of benefits and harms. So this goes back to what we've just been discussing. If you've got very little harms, then you may be more prepared to make a recommendation based on lower quality evidence. But if you're going to recommend major surgery or a dangerous drug, a lifelong drug to somebody, you want pretty solid evidence of its benefits, and that the benefits outweigh the harms before you would recommend it.
And I think the mother's kiss would fall into that category of something that doesn't have significant kind of obvious harms associated with it, and therefore is something that you're more likely to be able to recommend than something that actually has very clear potential for complications. And I think also it just highlights that actually one of the reasons why we need randomised trials is that looking at a single patient, in the vast majority of cases of things that we do in medicine, it's unclear whether it was that intervention that led to the outcome, or whether it was just due to chance or due to a natural progression. Whether it's you cannot say that if you have not had a heart attack 5 years after having a statin, whether that was linked to having the statin or whether you'd fallen into that category anyway. Whereas you can say in the case of an alligator that your arm probably wouldn't be attached at the same time.
That's right. And in general, the further the risk is in the future, like the risk of a heart attack, those sort of preventive manoeuvres, the harder it is to tell that your intervention has worked.
I'm aware of time, but I wanted to finish on a final question, which has been a bit of an interest in mine recently, surrounding medical education. And I was wondering, what improvements would you like to see in the education that we give people, that we give medical students, clinicians, about interpreting evidence or carrying out research? And what would be your main salient points here?
So that's an interesting debate currently within the evidence-based medicine community, is how much do you need to understand about the nature of research? And an analogy that I use for this is it's a bit like chest x-rays, right? You don't have to be at the level of a radiologist, but you have to understand what a chest x-ray is and what it's for. And you need to be able to interpret the report on it. So that would be the sort of, if someone gave you a report on a piece of research that said, well, this was biased because it was unblinded and had very poor follow-up, you need to understand those terms, because that's like reading your radiology report. And occasionally on a weekend in a casualty or something, you may find yourself having to read your own chest x-ray, and at least having some crude understanding of it is necessary, I think, for all practitioners. But you don't have to be super sophisticated, but you have to understand the importance of good research, and you have to have some basic understanding of the elements of it in order to recognise that this is a reasonable report on this research.
I guess I think we've talked about it on the podcast a little bit before, that perhaps in a more ideal world, you would need to understand less, because we would have very good guidelines in place, very good unbiased guidelines and sources of information, whereas actually there are times when you do need to interpret things for yourself, and that is not done as effectively by others as it should be, to be cynical perhaps.
Yes. And there's so much evidence that it's actually very hard to always have the appropriate guidelines for things. It reminds me of in my general practice once, when the Women's Health Initiative trial was published, which was the one that overturned the view about hormone replacement therapy for menopausal women. It hit the news, and we were getting deluged with phone calls for women wanting to come in. So we held an emergency journal club because we had the guideline. It actually took them about a month to take the current guideline down. That was their response of the authorities then. But we had to answer the questions in the meantime, and there were lots of voices over the radio and newspaper claiming different things and interpretations. So we thought we need to sit down and just go through this primary evidence ourselves in our emergency journal club. And that's like my analogy of having at least being able to read at least a basic, an obvious chest x-ray finding that you could see that this was a pneumonia or a severe heart failure, for example. Every clinician should be able to do that. And we were able to then do that with that particular piece of research, the JAMA trial.
Perhaps it's a naive expectation, but we do get taught a lot of things in medical school and in medical education that are preparing us for things that never happen, or diseases that we never see, specialties that actually we don't have much contact with and we just refer to. But even if, for example, you teach it to people and only 10% actually end up using it in, for example, conducting their own research, or only 20%, then that can still be seen as a net benefit for the system overall, compared to if nobody has any understanding of evidence-based medicine. The bar is not everybody being able to use it necessarily.
Yes, but everyone should understand it. For me, it's like, not everyone has to be a microbiologist or a radiologist, but you need to have some fundamental understanding of the different types of organisms or the different things that you will see on different imaging studies. That's sort of fundamental, and it takes a bit of work to do that. And it seems to me that the skills for evidence-based medicine that you want is not to reach the level of a microbiologist or a radiologist, but that you need some fundamental understanding and some skills, a little like using a stethoscope, for example, that you need an understanding of what it's doing and then some skills in doing it, even if you don't become a cardiologist. You still want to be able to hear a murmur. So I would see evidence-based medicine in being that everybody is going to need some basic level of understanding, no matter what your specialty is, because you will get deluged by information. And so you have to say, is this credible or not? What did they base it on? Was that a controlled trial? And having that fundamental understanding of the different types of research, their purposes are, and what would be adequate research, I think is a fundamental base across all fields of medicine. It's just that you then need a small percentage of people who are then highly skilled in being able to pick apart the funnel plot of a systematic review or interpret the I-square statistic and all of that stuff. Not every clinician needs to understand that level, but we do need a small percentage of people who actually do understand that you can then refer to and say, look, I can't read this piece of research. Can you come and give us a hand in interpreting this research? And we've been doing that for the local clinicians here for the COVID research, for example, in helping them with a COVID journal club, because some of the stuff is just beyond their level of skills. And so we've been the sort of the clinical epidemiology specialists for their interpretation of this stuff in the same sense that you'd get a microbiological or radiological consult on something.
Exactly. And perhaps knowing what you don't know and knowing what you can't do and what you can do is also quite important in the same way that it is across medicine.
Exactly. And you need enough knowledge to know that you don't know.
And you can't just read the final sentence of an abstract and get all of the information out of it. I think that would probably be, I'm aware I've taken up a lot of your time already. I think that would probably be a good note to end on for today. So I'd just like to say thank you very much for what's been a fantastic conversation, and I hope you've enjoyed it.
I have, George, and all the best with both your clinical and your research careers. It sounds like you've got great interest in both.
Thanks for listening to the Pager podcast. If you've enjoyed this episode, then do share it with a friend and come back to listen to our other episodes. As ever, we'd love to hear your feedback, and you can reach us on social media at pager_podcast, and you can also email us at pagerpodcast@gmail.com. We hope to see you again.